Hello everyone. So today I'm going to be talking about compilations of research done by Li, He, and Jiao and their findings in the updated review of the epigenetic mechanism underlying the pathogenesis of age-related macular degeneration. I know that sounds like a mouthful, but the underlying question at hand is fairly simple. But first, I want to go over some definitions. Epigenetics is the study of how behaviors and environment can cause changes that affect the way genes work. Epigenetics has been recognized to play an important role in physiological and pathological processes of the human body. The science behind epigenetics refers to the modification of DNA by external factors which turn genes on or off. This results in a changed function and behavior of the organism. This does not change the DNA sequence itself, but are heritable, dynamic, and reversible. Pathogenesis is defined as the manner of development of a disease. AMD, which stands for age-related macular degeneration, is defined as an eye disease that can blur the sharp central vision you need for activities like reading and driving. The macula is the central area of the retina. In this region, the macula is seen as the dark figure in the center of the screen, which is notated by a blue arrow. The light circle, which is shown to the right, is the optic disc. This is where the optic nerves form. Macular degeneration is the deterioration of the central portion of the retina, the inside back layer of the eye that records the images we see and sends them to the optic nerve from the eye to the brain, is diminishing. The retina's central portion, the macula, is responsible for focusing central vision in the eye. When working properly, the macula collects highly detailed images at the center of the field of vision and sends them to the optical nerve of the brain, which can then be interpreted as sight. Age-related macular degeneration is considered incurable and is the leading cause of blindness in the elderly in developed countries. It affects more than 10 million Americans to this day. When the cells of the macula deteriorate, images are not received correctly. In early stages, macular degeneration does not affect vision. Later, as the disease progresses, people experience wavy or blurred vision. If the condition begins to worsen, central vision could be completely lost. People with AMD are deemed legally blind but retain their peripheral vision. This picture represents a moderate to severe form of AMD. So with those definitions in hand, the true question we will consider when moving forward will be, how do certain behaviors and environments play a role in the development of the disease known as AMD? Despite the fact that there is currently no single animal model that develops all signs of AMD, animal models are necessary for the study of epigenetic mechanisms. The mouse is usually used in correlation to human-related studies due to versatility and previous knowledge. Twin animals are also often used when it comes to epigenetic studies. Since the twins have identical genomes, identical twins are the ideal subjects for studying modifications. Genetically confirmed monozygotic twins can present initially with similar early macular pathology. However, during the late stages of the disease, physical traits can differ in terms of the time and specific type of disease. In some cases, one twin can develop the disease while the other does not. Although there is still information to be obtained, Epigenetic mechanisms such as DNA methylation and histone modification may provide a new understanding of the pathogenesis and provide a potential therapeutic approach for treating this complex disease. DNA methylation works by adding a methyl group to DNA. When added to specific places on the DNA, it can affect the DNA-protein interactions. During methylation, the gene is turned off, and during demethylation, which means to take the methyl group away, the gene is turned on. Histone modification is another epigenetic mechanism. This is when DNA wraps around proteins called histones. When they are wrapped tightly, the gene can no longer be read. Therefore, when it is wrapped, the gene is off, and when it is unwrapped, the gene is on. Inflammation in the macular area is known as the leading cause of AMD progression. In one study for certain individuals, relevance of DNA methylation to AMD pertains to a gene encoding protein. This protein is associated with clearance of cellular debris and contains a special area in the promoter region that could have anti-inflammatory functions. Increasing the methylation of this protein was confirmed detrimental for AMD sufferers due to increased inflammation. Hypermethylation, meaning too much methylation, was identified and associated with increased expression of the particular gene in patients with AMD. In this updated review, it was also notated that histone modification changes chromatin structure by recruitment of remodeling enzymes and forming a complex to influence cellular function. Identifying the reader proteins of histone modification was also a detrimental factor in confirmation of AMD sufferers. It was reported that significant increases in messenger RNA is associated with excess levels of iron. This leads to relation of increased iron as a risk factor of AMD. To summarize, age-related macular degeneration is a very complicated disease mediated by multiple factors including genetic and epigenetic factors. Much attention has been paid to the contribution of genetics and epigenetics to AMD pathogenesis. However, how the interplay of genetic and epigenetic factors affects the development of AMD remains unclear. DNA methylation and histone modifications are epigenetic modifications that can be introduced over a lifetime. Looking into these mechanisms provide better clarification on what is happening at a molecular level and how it affects gene function and overall the organism. Currently, there is no cure for AMD. The most common treatment method of AMD is the injection of anti-cancerous reagents. However, complications are seen within the eye and systematically. Individual responses to the injection differ and repeated injections are needed, which often promotes many other challenges. Because epigenetic modification is dynamic and individual specific and AMD is a complex disease, further research is needed in these directions. However, in the last decade, all of this progress has been made. This leaves some hope for a cure for age-related macular degeneration.